Now that you have an understanding of the mechanisms behind the phage display system, let's have a look at how it's used in research. Phage display has a broad spectrum of applications, which can go from improvements of enzymes to proteome analysis or drug and target discovery, but is certainly not restricted to these. The first thing I want to focus on is optimization of enzymes. A large variety of enzymes are being used today in different fields of industry and biotechnology. These enzymes are originally made by organisms for a specific function in a specific niche or environment, like inside the cell cytoplasm. Therefore, they often have undesirable properties for the function or environment we want to use them in. They might not work fast enough, be unstable in the conditions they are used in, or do not work on the desired substrate. Phage display can aid in improving these enzymes by selecting, for example, for better term stability, activity, or enhanced substrate binding. Let's start with stability. Take, for example, an enzyme that is normally present in a cell cytoplasm at 37 degrees Celsius. However, when we use it in an industrial process where we need 50 degrees Celsius, the enzyme becomes unstable and denatures. In phage display, we can make a library of mutagenized enzyme and display them on, in this case, the gene 3 protein of the phage. In the binding step of the biopanning, most phages will not be able to bind the enzyme substrate at 50 degrees Celsius, as they are denatured. Only phages who display an enzyme which is able to fold correctly at this temperature will be retained and amplified in the subsequent steps of biopanning. These phages can then be sequenced to obtain the sequence for the stable protein. Resistance against proteolysis can also be acquired through phage display. In this example, a library of mutagenized enzymes is expressed as a fusion protein with the enzyme of interest in the center of gene 3 protein. After digestion, only phages carrying a proteolysis-resistant enzyme have the N-terminal tail of G3 protein, which is required to interact with F pilus. Hence, only these phages are amplified in the biopanning step. The DNA sequence obtained from these phages will again encode this resistant enzyme and can be used for production of the modified enzyme. It is also possible to enhance catalytic activity of enzymes. In this example, I will discuss a way to do this for metalloenzymes. These are enzymes that require a metal atom for their activity. In this case, it was described for a beta-lactamase, which is an enzyme that is able to inactivate a beta-lactam antibiotics. A library of mutagenized beta-lactamase was prepared, fused to the gene 3 protein of M13. In the first step, the phages are treated with chelating agent, which removes all the metal atoms. This way, the metalloenzymes are inactivated. However, they are still able to bind their substrate. Hence, a first selection is made for enzymes that are still able to bind their substrate. After washing, the metal is added again so that enzymes which are able to function are activated. More active enzymes will cleave the substrate better and hence elute better. In this way, a positive selection is made for enzymes that are catalytically more active. After 2 rounds of biopanning, the researchers acquired an enzyme that has a 60-fold increased activity. The technique can of course also be extended towards other metalloenzymes. Phage display can also be used in proteomics research. Many proteins affect their protein function by interaction with other proteins. However, in many cases this interaction partner is unknown. To find these protein interaction partners through phage display, a cDNA or ORF library is made from the proteome of interest. These libraries encode practically all possible proteins that can be expressed in a certain cell. Hence, the different phages in the library will express and display all these possible proteins on their surface. In this case, gene 8 protein was used for fusion. Panning is then done by using the protein of interest as a bait. After a couple of rounds of panning, the remaining phages will be enriched in proteins that interact with the bait protein. Sequencing them will generate a list of potential targets which can then be verified by other techniques. The last example I will show you is the search for a peptide that can pass the blood-brain barrier. In drug development, it is hard to develop drugs that are capable of passing into the brain. This is because the brain has a so-called blood-brain barrier. This barrier is only present around blood vessels in the brain and not in the rest of the body. The endothelial cells of brain blood vessels are connected by tight junctions that form a continuous and hard-to-pass barrier between the blood vessel and the brain. This forms a problem in, for example, the development of drugs against Alzheimer's disease. However, the barrier is not impossible to pass. Some proteins and nutrients are able to pass through the barrier. One way to allow drugs to pass would be to link the drug to a small peptide which is able to pass. Hence, researchers set up a phage display system to find such a peptide. To this end, a peptide library of random 12-mers was constructed to be displayed on the phage surface. Rather than doing in vitro biopanning, an in vivo panning approach is used to find the peptides. In this panning technique, the phages are injected into the tail vein of mice, and the phage is allowed to circulate for minutes to hours to allow binding to the receptors. The mice are then euthanized and the phages are extracted from the brain. These phages are then used again in the next rounds of panning. This will give an enrichment in phages containing a peptide targeting the brain. After 4 rounds of in vivo panning, 20 peptides were eventually sequenced. 12 of them had the same consensus sequence. This consensus peptide was then fused to a nanoparticle and labeled with a fluorescent tag and injected into the mice. In vivo imaging was then used to look at the localization of the fluorescent nanoparticles. The first 2 mice are control mice, and the third and the fourth have the fused particles in different ratios. A clear localization to the brain region of these mice is visible in the third and fourth mice. Also, when you look at the localization in the organs separately on the right figure, you can clearly see a strong accumulation of the nanoparticles into the brain, which is not the case for the control mice. Hence, the peptide discovered by phage display could target nanoparticles or drugs to the brain. These examples clearly show the versatility of the phage display system and how they can be applied in practice.